we are lucky to be joined by Dr. Trevor Day. He's going to be talking to us about integration of respiratory and renal responses to acute and chronic blood gas challenges, some novel insights and future directions. Dr. Day is currently a professor of physiology in the Department of Biology at Mount Royal University just down the street. He did his Bachelor of Science in Psychology with a focus on neuroscience and physiology at the University of Calgary, and also did his PhD at the University of Calgary under the supervision of Dr. Richard Wilson on cardiovascular and respiratory sciences, and specifically respiratory neurobiology. So thank you for joining us today, and we're looking forward to your talk.
Thanks for having me. Thanks for Jared for reaching out with a panic text. Like, someone canceled, can you fill in? So thanks a lot, Jared. But really grateful to get the offer to come. I was here a couple of years ago at the invitation of Mike Asmussen, talking about brain blood flow in response to high altitude responses. Happy to come back and do something closer to my wheelhouse that I'm up to recently around respiratory renal interactions and acid-base balance. I pitched it broadly, so I'll try and catch us all up on the basics, so to speak, of acid-base balance. But I want to tell you some stories about high altitude descent along the way. So thank you everyone for coming. It's great to see lots of familiar faces. And this is my alma mater, even though kinesiology isn't, but both my degrees are from UC. So it's a bit trippy to walk around campus today and have lots of strange old memories. So cool to be back. So this is my visual abstract that I got a student of mine who's also an artist to do. This took like 10 napkin drawings. And then she put in the windmill and the water pump as kind of a little her idea, but this is inspired by Wasserman's wheels, and we'll come back to that in a second. Really, my talk, it's just working. I tested it 1 minute ago. Now it's jammed. Stand by. I just tested it before I started talking. My apologies. Okay, so my talk really should be carbon dioxide is the exhaustive lake, because it is. It is a metabolic waste product that has profound acid-base implications. And if we didn't breathe to get rid of it, it's a volatile acid, but also have kidneys to help compensate when breathing goes wrong or right, as you'll see. We would have a catastrophic acidosis, and that's what we're here to fix today. And so as all the PIs in the room know, we're nothing without our collaborators. We're nothing without our funding agencies. Of course, we have to play nice in the ethic board sandbox as well. And we're nothing without our collaborators internationally as well. The data I'm going to show right at the very end today is actually in review right now with one of my students leading it. So we're waiting to hear news. It hasn't been rejected yet. But it comes from a Nepal expedition that we carried out in 2023, led by Professor Tom Broussard, who's from Syracuse University, and Pontus Holmstrom, who was a postdoc at the time at Mid-Sweden University, and coordinated with one of my favorite people in Nepal, Nima Sherpa, who I've worked with 5 times in going to do expeditions in Nepal, and the entire Nepal 2023 research group. This is us in a village called Farishay at 4,300 meters in along the Everest Base Camp trek. And we just finished about 5 days of research in this cafe/lab, and we're about to head up the hill to Everest Base Camp from here. And so a large gratitude to this group who helped put this expedition together, some of whom volunteered to be participants. And so this is in collaboration with UCLA and Syracuse, who helped together, worked on this NSF grant that we landed a couple of years ago. I'm also funded by Insert Discovery Grant, and of course Mount Royal, helping with some funding and equipment along the way. And then we had to play nice in the Nepal ethics sandbox as well. A big shout out to undergraduate students. So I'm at Mount Royal, and Jared knows this all too well. I work primarily with undergraduate students. Sometimes I work with advanced trainees from other places. I somehow find ways to work with postdocs at other universities. So I'm lucky to have that help. But my research program is largely run with the help of undergraduate students. And so even though this isn't Nepal, this is the most recent picture of our group at Lake Titicaca, Bolivia. Jane was there with us recently in July. Paolo couldn't come, but we tried, but he sent some students along. Rodian just started medical school in Calgary. Jessica just started a master's in my own lab. Manal is here, I believe, with Brittany. I'm not sure if she's in the room. That's a scolder for not showing up. Nicole just started an after degree in nursing at U of A. Ben just started a master's with Craig Steinbach in Edmonton. And this is next to Ozakowski, who some of you might know, who I collaborate closely with at Mount Royal. So, undergraduate students are smart, ambitious, work well in teams. Those that want the research endeavor as part of their training are really hungry, and I'm grateful for the ability to work with them along the way. So many of you know a little bit about Mount Royal University. I've been there for 17 years full time. I don't know where the time went. You'll snap your fingers and your life will be over too. But I joined in 2007 after my PhD over at UC Medicine. Mount Royal is about undergraduate baccalaureate granting institution only. It's about 13,000 students, maybe 10,000 full-time equivalents. And it was a transfer college for most of its existence since 1910 to 2010. I joined right before we became a baccalaureate granting institution. I snuck at the professoriate through the back door. And so this is the campus that I work in. But because of the context that I work in, I have my fingers in a lot of pies. And so perhaps one might criticize me to say that my research program is not well focused, and you're probably right. I started the integrated physiology lab in about 2010 or 11 or so to integrate our teaching and research together. I trained in animal physiology, comparative animal physiology, and so didn't really have much of a background in working with humans, the intact human model system, although I was an EMT in the previous life. And so this lab was utilized to start building up our teaching and research capacity. Here I am working with a bunch of students in 2017, doing baseline testing, about to head to Nepal for our 2017 expedition, data from which I will show you today. But I play in a lot of different sandboxes. Sort of my bread and butter and where I was trained is in chemoreflex control of breathing, responses to blood gas stressors. I've started to, since about 2012, when I went to Nepal for the first time, started dabbling in high altitude hypoxia and acclimatization. About a third of my program has been brain blood flow regulation, although I'm moving a bit away from that. And then lastly, I'm really interested in lots of integrative responses. Certainly got some work with central sleep apnea that if I had time at the end, I might show you some data from Bolivia, but largely going to focus on renal acid-based responses in response to chronic respiratory challenges like being at high altitude. So people ask me at cocktail parties, and they don't, what do you do for a living? I say, well, I'm an integrative cardiorespiratory, cerebrovascular, and blood acid-based physiologist. Don't ever do that. Really what I say is I'm interested in how the heart, lungs, brain, and kidneys talk to each other in response to stress, and my favorite stress happens to be high altitude, at least recently. So here's a picture I took of Everest. Kidding, of course. It's taken from the south side. I've been here 5 times, I'm happy to say. I took the environmental physiology course, by the way, in 1998 here with John Cole. Does anyone remember those days? That Reading into Thin Air by John Krakauer in that course with John is probably why I'm standing here today. That course and John himself inspired me to become an environmental physiologist. And so we read and dreamed about going to the Everest region since back then. In 2012, as I scrambled up a little side summit here called Kalabatar, I couldn't believe that my dream had come true to be in the Everest region. This is the Nipsey Wall that kind of separates the south from the little bowl where the Everest Base Camp trek and the Khumbu Glacier goes. Nipsey Wall, Lhotse Face, there's the top of Everest. The highest terrestrial point on Earth. John West, the grandfather of high altitude physiology, famously said that if Everest was any higher terrestrially, we probably wouldn't be able to climb it. My question to John would be, well, who doing what? You know, a pregnant woman, a Sherpa, like who are we talking about here? But it's true that it's a profound stressor, and even Sherpa use oxygen above 7,500 meters as they head up to this highest point. Climbed first in 1953, and now it's a bit of a disaster in terms of tourism, but it still generates stories and dreams of doing wonderful things. This little pyramid-shaped mountain here is called Pamori, and it's kind of in the shadow of this little lab we went to in 2017, so 2016, 17, and 18. I'll probably show you pictures of it along the way. But you just basically go up the Khumbu Glacier, go around this corner into this bowl to the Everest Base Camp trek, which ends where the Everest climb and the ice falls begin. Some of you probably know about Wasserman's wheels, and this is my small little throwing you a bone to exercise physiology, and it ends after this slide. But I actually gave a version of this talk at UCLA at the Lundquist Institute in the shadow of Wasserman and Wick, so it was pretty trippy to put this up, knowing where I was. But I like this model of trying to understand how the lungs the heart, the blood, the muscle, and the metabolic rate all work together integratively to try and serve the metabolic rate during exercise stress. And this was on my mind when I was thinking about the integration of respiratory and renal interactions in maintaining acid-base status. And so based on Wasserman's wheels, I sketched something out like this, and Val and I were sending, text back and forth with pictures on napkins, and finally, we think we got it right. We've got the lungs here, the heart and blood here, the kidneys there, the metabolic rate, which is the site of CO2 production and oxygen consumption, the windmill here, driving oxygen consumption and CO2 elimination. So we'll talk a lot about that part today. And then the kidneys with filtration and either secreting hydrogen ions or reabsorbing bicarb along the way to maintain acid-base balance. So I call this my visual abstract for where we're going the rest of today. So where are we going? Here's my sort of overview. I'll come back to this after each section so you can go, oh, thank God we're at 7. It's over soon. But we'll start with a little background about respiratory chemoreflexes, which is the topic of my PhD where I first trained and sort of my starting point for where this all came from. So I made this sort of schematic diagram, this sort of really textbook level for a review we're working on right now. We know that the respiratory controller, the clock that ticks that makes you breathe 12 times a minute, is in the brainstem. We've known since the 20s in cats. We know that there are neurons that impinge upon respiratory muscles to control alveolar ventilation, and changes in alveolar ventilation affect blood gases, and you can thus change CO2 and O2. There are sort of normal values at sea level, and those changes in blood gases, which can be imparted by either breathing, hyperventilating or breath holding, or changes in the fraction of inspired oxygen levels if you're in a laboratory doing acute studies. Both of those ways of changing blood gases are detected by 2 sets of respiratory chemoreceptors. These are neurons or modified neurons that taste blood gases in the same way that your tongue tastes glucose. And so these peripheral chemoreceptors or carotid bodies are at the bifurcation of the common carotid artery. It's just under the angle of the jaw where the common carotid body artery splits, and that little group of cells taste blood gases on the way into the brain. They're quite quick, sort of a breath-by-breath maintenance of changes in O2 and CO2, but they have a synergistic effect. There's a stimulus interaction between O2 and CO2 in terms of their control of breathing on a very rapid time course. There's also another set of chemoreceptors found throughout brainstem tissue. There's multiple sites of them actually. The best described are on the ventral underneath surface, but they're buried in tissue. I know your textbooks say they taste cerebrospinal fluid, but that's not true. They're really buried in tissue, and they taste changes in CO2. Likely intracellular hydrogen ions are what they detect. But because there's a temporal delay, as well as an equilibration delay between blood and brain tissue, they're a lot slower. But both sets of chemoreceptors impinge upon the respiratory controller. The topic of my PhD was whether or not they talk to each other and interact, and that debate still goes on today. And we're still fighting that debate actually. But ultimately they can impinge upon the respiratory controller and around we go in a negative feedback loop. So the response to this, we call it the gain or the responsiveness or the sensitivity of these systems in response to challenges. My old supervisor, Richard, and I are still working together. We made this little schematic a few months ago as part of a commentary, but I thought it was a nice way of just thinking about how people respond to blood gas challenges in an acute level or more chronically. So we can think about acute responses. I can bring you into the lab, make you breathe low oxygen. Jared and I do that all the time in Mount Royal, and we can measure responses in seconds, minutes to hours with time-dependent effects as well. If you were to stay longer at that stimulus, usually now going to high altitude, but not always, you can do chamber studies. Not many have been done, but some have been done on a longitudinal basis, and we can acclimatize, or in a lab perspective with more isolated stimulus, we can call it acclimation, but we can measure that over days to weeks to years. If you grew up at altitude, Paolo had a twin brother in Vancouver, and one of them stayed in Vancouver, became a yoga instructor, and the other ended up going to the Andes and grew up there. They would look different as adults, and so that developmental adaptation that takes place over when you grow up at altitude, you look different, and that's not reversible. So that's the beginning of the irreversible changes. And lastly, if you spend generations, if your ancestors spend generations at altitude, as is the case with Tibetan Highlanders or Andean Highlanders or Ethiopian Highlanders, you will get genetic adaptation. So this is evolution in our species. So we have a couple of examples of this in various populations around the world, but this is across generations. So if you want to study high altitude indigenous or native high altitude people, it's very difficult to know that the effect that you're seeing is it just long-term acclimatization because they live at 4,000 meters? Is it developmental because they grew up there? Or is it genetic because their ancestors are from there? It's very difficult to know. The best studies probably are to bring them down to sea level and watch them deacclimatize, but those are incredibly expensive and difficult logistically to do. So often what we do is we go to high altitude and we study them there, which of course has all kinds of caveats. I'm going to show you some data today when we recruited Tibetan Highlanders or people of ancestry from Tibetan Highlanders at low altitude and went up with them together. And very few studies have done it that way. So let's start with acute responses. This is a picture of the carotid body, which is our main hypoxia or low oxygen sensor at the bifurcation of that common carotid artery. It's got a small nerve that goes to the brainstem. And as you reduce alveolar or arterial or end-tidal O2, you can see that you get this, this increase in ventilation that really ticks off below about 60 torr or so. And so that kind of classic hyperbolic response was characterized actually in the 50s in humans, and we've known about it ever since. If you're an analysis wanker like me, it's very difficult to quantify, or it's harder than linear responses. But this is the sort of typical response in ventilation to drops in oxygen. You can do steady state tests in a lab. This is us in our lab doing some steady state tests from Douglas bags. Here's a tracing from Craig Steinbeck's study in Mark Pulman's lab in 2007. I was probably a participant in the study because that was next door to me back in those days. But you can using an end-tidal forcing system, you can drop oxygen as the stimulus, hold it for 20 minutes. You can quantify a respiratory response. You know, there's a roll-off, and you look at that peak response indexed against the stimulus. Because when you breathe more, though, you drive carbon dioxide down. And that's called poikilocapnic hypoxia. And so in this case here, you can either clamp CO2 and keep it flat, and you get a very robust response. But if you let CO2 float and the CO2 drops, because as soon as you start breathing and CO2 are inverse to each other when the feedback loop is closed, and you can see that CO2 is driven down, and that of course blunts the respiratory response as well. So by the end of 20 minutes, you're not really breathing that much more than you were at baseline. In fact, I can tell you, having measured resting ventilation, even at 5,000 meters, people aren't breathing that much more than they are at low altitude, largely because, yeah, they're hypoxic, but they're driving their carbon dioxide down. Because of that stimulus interaction with the carotid body between low O2, but also changes in CO2, you actually blunt the hypoxic ventilatory response, and that's what we see here. So that's a steady state, but acute response that you can quantify in the lab. So if you want to keep the feedback loop opened up and you actually clamp different levels of CO2 in that same kind of method I told you about a minute ago, this is original data from the 50s that I plotted. You know, increases in CO2, nice linear response in ventilation, contributions for both central and peripheral chemoreceptors. From an oxygen standpoint, this is from Weil in the 70s. I've shown you this already, a hyperbolic response. But if you index it against oxygen saturation, it linearizes, so it feels a lot better to quantify slopes in this case. And so you can see the sort of standard integrative human responses to challenges in blood gases. If the feedback loop is closed, that is to say in normal situations, and you're to change your breathing voluntarily, you can hyperventilate. And when you hyperventilate, that's great in terms of oxygenation. You bring your oxygenation up, and if you're in a high altitude context and you hyperventilate, you'll bring your oxygenation up and you'll protect yourself. But if you were to hypoventilate, underbreathe, the most extreme example of that would be breath holding. If you hold your breath, you accumulate CO2 at the metabolic rate, CO2 starts to go up. And so this is, you know, interesting to know this, that when you're at altitude and you elicit, or even in the lab, the hypoxic ventilatory response, you protect oxygenation to some extent, but the unintended consequence of that is that you get low CO2 or hypocapnic. I'm going to use that term a lot today. And acutely at least, and that's the time course we're worried about, is you get a respiratory alkalosis. That is an important acid-base disturbance that your kidneys have to fix, because you're not going to bring your breathing back down. You have to maintain it high pressure to protect your oxygenation at altitude, but the CO2, the hypocapnia causes an alkalosis that the kidneys now have to fix. So let's now move out of the lab and head up the mountain to high altitude and see how we acclimatize. So just in terms of background, if you're curious why high altitude is a stressor, the atmosphere literally thins as you ascend. The atmosphere is a fluid just like water is, it just happens to be 1,000 times less dense. Gravity acts upon the gas molecules though, so at sea level, there's a column of air above me that's exerting a pressure based upon gravity. Well, similar things happen when you dive, and you're probably more familiar with this. For every 10 meters you go below the surface, another atmosphere gets added to you. And you feel that pressure a lot more because it's 1,000 times more dense than air. So it's the same idea that as you descend, the column of water molecules above you is larger and gravity is acting upon it. It's the opposite effect when you go to high altitude. So as you ascend, the atmosphere density thins. The fraction of inspired oxygen is 21 here. It's also 21 on the top of Everest. But because 21% of less atmospheric pressure is less, you then have a relative hypoxia in the atmosphere. So here I put this in a textbook years ago, this increase in altitude there in kilometers now, and then gas pressure on the y-axis. You can see the overall atmospheric pressure from sea level at 760, that's in Vancouver. And then from there, you can see in Calgary, we're about 1,100 meters or so. You can see we're more like 660, 665, something like that. And then 21% of this is this line, and you can see we become hypoxic. So Vancouver sea level, Calgary's about 1,100. We pressurize an airplane cabin to about the equivalent of 2,400 meters or so. So you are mildly hypoxic in an airplane cabin. And then all the way up to the top of Everest at just below 9,000 meters or so. Coincidentally, where I'm going to take you today, at 5,300 meters in the Nepal Himalaya, happens to be about half the available oxygen at sea level. So highest city in the world, Cerro de Pasco in the Andes, or Everest Base Camp is about half the oxygen of Vancouver. So it's a wonderful place to go to study hypoxic stress here. This is from John West's excellent but straightforward textbook on the oxygen cascade that I've redrawn here. And it's helpful to think about the source of oxygen and the sink for oxygen. The source for oxygen is the atmosphere. At sea level, it's about 160 torr or so, 21% of 760. And then as you head down to the alveoli, we're now diluting some of that oxygen down with other gases, water vapor, carbon dioxide, that kind of stuff. You get down to about 100 or so in the alveoli and arterial blood, even lower in tissue capillaries, lower still inside cells. And lastly, if you stuck a PO2 electrode inside a mitochondria, you'd get between 2 and 5. So just like glucose is low inside cells because the metabolic Pac-Man is chewing it up, oxygen is low inside cells as well for the same reason. So you're kind of on a knife edge here, aren't you? Even though there's an abundant amount of oxygen, air breathers have this advantage of having an abundant amount of oxygen. We breathe in 25, maybe we breathe out 15. We don't use very much. Even venous blood is 75% saturated with oxygen. So we've got lots of extra, but when you get down to the mitochondria, it's actually quite low. So if you start lower, because of your atmospheric level is lower when you go to high altitude, you can see that this cascade then has to accommodate in some kind of way through ways that I'll tell you about in a second. So let's talk about acclimatization on this continuum or time domains of this. This is from Phil Ainsley's excellent review from over a decade ago. You can see that transient hypoxia-ventilator response I showed you before. It rolls off, but over time, days to weeks, because the carotid body gets more sensitive, because of hypertrophy and hyperplasia, those cells become more sensitive to a given stimulus. Your baseline breathing goes up. And I think the baseline breathing is actually more important than any transient response, but we can fight about that later. But you can see baseline breathing goes up over time. We call that ventilatory acclimatization. I drew this little thing. I should probably get an artist to help. This is a garbage overview, but I think for the undergrads or grad students that are interested in high altitude, it's a nice place to start as an overarching theme of what acclimatization means. People often drop that word, but they're not specific about what they mean. Do you mean renal and bone marrow and hematopoietic acclimatization, making more red cells? Do you mean increased sensitivity of the ventilatory control system to breathe more? Do you mean acid-base balance? And these are different types of acclimatization that happen on different time scales along the way. And so in response to a hypobaric, that is to say low atmospheric pressure from ascent to high altitude, hypobaric hypoxia, you get both an acute and chronic hypoxia-ventilator response. Acutely, it's very variable between people. Chronically, it becomes more sensitive over time, also still very variable between people. But also you get a renal erythropoietin release, and this is a hormone that the kidneys release in response to low oxygen in renal blood that releases it into the bloodstream, acts on red bone marrow, increasing hemoglobin mass, measured indirectly often with hemoglobin concentration or hematocrit, but the gold standard would be hemoglobin mass. Martin helped me with some of this in 2023 in Nepal. Still grateful and won't forget in terms of some gear that we needed. And then because of that, and because breathing protects oxygenation and protects oxygen saturation, A here means arterial, together through some math, you get an increase in oxygen content. So you can protect your oxygenation through two sort of pathways that interact together, both with lots of variability. But also, the unintended consequence, as I said, of breathing more causes low CO2, causes a respiratory alkalosis, at least acutely, which has to be fixed. You're not gonna back off on your breathing, because you need this, right? And so because of that, on a longer time course, the kidneys will then excrete, not secrete, they leave it infiltrate, because you really filter it, and then you'll just leave it there and excrete it into the external environment, and you get this compensatory, I would call it a relative metabolic acidosis. You're not actually acidotic, but you're relatively acidotic, so you bring your pH back to normal. So as my PhD student Jack Lacey in Ireland often says, going to high altitude and studying these responses is like spinning plates, because there's all these different organ systems that are responding, sometimes appropriately, sometimes not appropriately, which could lead to altitude illnesses. Acclimatization may not be helpful. You can overdo these responses, like the Andeans do when they make truckloads of red cells and get hematocrits of like 70%, and their blood is super viscous, and they get chronic mountain sickness is one example. But you see, you're spinning plates, and any response from one organ system becomes a stimulus for the other. And if you drop one plate, the whole thing can cascade out of control, and you can develop altitude illnesses. So for the rest of today, I just want to focus on this half over here and show you some data from some of our expeditions. The topic of my talk was about integration between the respiratory and renal systems, and so let's talk about that again really briefly. We know that the hypoxia-ventilator response protects oxygenation. That sounds like a good thing, but you decrease PaCO2 and you get at least acutely a respiratory alkalosis, and you're off the pH scale. But on a longer time course, if this is maintained, then the kidneys can actually control both bicarb and hydrogen ions together. We call these metabolic compensations, and that can bring pH back down to normal again. So what did we learn? We know that an HVR in response to hypoxia drives an increase in ventilation, drives a drop in CO2, causes a respiratory alkalosis. Longer duration exposures sensitize the ventilatory control system, so the hypocapnia gets even worse over time. And so the kidneys can then kick in, dump bicarbonate in urine, and dump it out here. Normally you secrete only hydrogen ions and bring back all the bicarb, but in this case, you drop some more bicarb into the stream here, the urine stream that Bell drew. And hopefully, but not always, depending on the altitude, restore pH and metabolic function. Who cares? This is a little bit of chemistry to catch us up. I considered hiding this slide, but I thought, well, maybe I'll just run through it quickly to make sure we're on the same page. Acid-base physiology really is the study of a proton, a single hydrogen ion, not bound to anything, like say hemoglobin molecule, but free in solution. And if it's free in solution, it can bind to proteins and enzymes and affect the shape of those proteins and enzymes. And ultimately, that's what serves the metabolic rate at the biochemical level. pH isn't a thing. It's a data transform of a thing. We just think 40 nanomolar is not very precise. Can you imagine 40 times 10 to the minus 9? Like, it's very difficult to imagine that kind of concentration of hydrogen ions. So we do a logarithmic scale and call it pH instead. 7.4 feels like a cozier number to deal with, don't you think? A buffer, like a sponge, is any chemical that resists changes in pH when you add protons to the solution. The main buffer in our physiological systems is bicarbonate, but we can dissolve CO2 in water and make bicarbonate. And that relationship between CO2 and bicarbonate is the most important buffer pair that we have. But the important thing here is that enzymatic reactions can be negatively affected or positively affected by changes in pH, at least outside of optimal ranges. Just to give an example, this is a three-dimensional view of hexokinase, the first of 10 glycolytic enzymes that changes glucose into glucose 6-phosphate in the metabolic rate in glycolysis. That 3D shape, the quaternary structure, can be affected by free protons. And so having hydrogen ions outside of the normal range can affect that 3D shape. Another one that you're all probably familiar with is that the shape of the oxyhemoglobin curve. Across PO2s, the amount of oxygen butts in hemoglobin seats can change depending upon both the pH, but also the CO2 independently as well. So pH really matters in terms of the shape of proteins for optimal function. This is a couple of examples. It's governed by this Henderson-Hasselbalch equation. Well, this is the carbonic anhydrous equation, but I'll cover this in a second. So the lungs and kidneys collaborate on acid-base balance. Whenever we talk about CO2 changes, call it respiratory. Whenever we talk about bicarb and hydrogen ions, call it metabolic. I know it's kind of an awkward term when we think about metabolism in response to, say, exercise stress, but these are called metabolic changes. And so CO2 in water, where's the hydrogen ion? Why is that called an acid? Well, it's because it combines readily with water to make carbonic acid, which doesn't really exist. It immediately dissociates into hydrogen ions and bicarbonate. And this is supposed to be red on my screen. This reaction here is facilitated a millionfold by the enzyme carbonic anhydrase, which you find everywhere acid base is important: stomach, pancreas, glia in the brain. The tongue for tasting carbonated beverages, by the way. If you take a carbonic anhydrase blocker like acetazolamide, you drink beer and it tastes flat. A little fun fact for parties. So whenever you go to high altitude and you're taking Diamox, you'll be like, what's wrong with this beer? No, it's you. You block the acid sensing in the tongue and buccal mucosa. But just remember that CO2 is carried largely. Some of it's found in hemoglobin, some of it's dissolved in water. Once it's dissolved, you find it in bicarbs, because the CO2 buried in here. And so CO2 is largely carried as bicarb in blood. So changes in bicarbonate, if you witness this, if you observe this with blood draws, it could be because they're just breathing more and driving the CO2 down. There's less CO2, there's less bicarb, but also it could be because the kidneys have jumped some as well. So getting at that time course and the magnitude of those differences is important, and I'll show you some data in a second about that. So here's the Henderson-Hasselbalch equation. Don't worry about it too much, just to know that pH is directly affected by bicarb and inversely affected by the amount of CO2 around. So if CO2 goes up, that means pH goes down, acidosis. If CO2 goes down, like you're breathing more, pH goes up, or alkalosis. There's normal values, and I do want you to memorize this one thing. It'll be a quiz after. Normal pH is about 7.4 with lots of variability, but textbooks say you are all 7.4 right now. And that is mediated by having a bicarbonate of 24 millimolar in the blood and a normal CO2 of 40. We're already lying to you, because in Calgary, I can tell you from measuring it, where poor Paolo just moved here from a lower altitude, and so it's gonna screw up our lab, screw up our data, because now you're sitting more like 36, 34, because we're at altitude. And so even that alone, being in Calgary alone, perturbs the system a bit. One slide on cellular stuff, and I'll promise to move on. So this is a renal tubular cell. This is the tubular lumen. This is where filtrate is made and dumped. I don't know what the hell happened there. But along the way, you freely filter bicarbonate. So this is really important to me, that GFR, a normal filtration rate, normal glomerular filtration rate in the kidneys is like 180 liters a day. That's a huge amount of water that you're filtering. Now, you're probably doing the math in your head. We've got 5 liters of blood, maybe 3 liters of that is plasma. How is that possible? It's possible because we virtually reabsorb all of it. But we need to filter it first and then selectively secrete and or reabsorb along the way for this filtrate to graduate and become urine. But when there's 24, supposed to memorize that, that's like over 4,000 millimoles per day of bicarb. If you didn't bring that home, you would die of a catastrophic metabolic acidosis. For every hydrogen ion you're trying to get rid of as a metabolic waste, you'll reabsorb in a complex kind of way one bicarb molecule. It's freely filtered. Luminal facing carbonic anhydrase splits it. Intracellular carbonic anhydrase brings it back again, and then basically it's kind of the same bicarb that gets reabsorbed. So, and here's the thing: the pH of this filtrate and the presence and amount and abundance of carbonic anhydrases can all affect how much bicarb you bring back or leave in the filtrate, and that's the topic of the rest of the talk. I need to lastly kind of orient you to a Davenport diagram before we go any further and show you data. This was from the 70s. Davenport created this diagram so that clinicians could quickly assess people's level of respiratory or metabolic primary disturbance and metabolic or respiratory compensation. And so here's our starting point. I asked you to memorize 7.4. I asked you to memorize 24. And I asked you to memorize 40. So we have these isopleths along the way, these span of CO2 curves. If we're starting here, and then we hyperventilate and drive our carbon dioxide down, then we have to stay along this non-bicarb buffer slope of about minus 22 or so. We head down to another CO2 isopleth. Now we're say at 20. That's a pretty profound hyperventilation, but easy enough to do, at least acutely. You'll feel a bit dizzy, but you drive down to here. So notice here that bicarb dropped a little bit, right? And that's not the kidneys. That's just the chemistry of it. And then from there, if you want to dump bicarb further to compensate, you have to stay on that isopleth because now you're breathing to bring down 20, and you dump even more bicarb, so you head down the y-axis, and that brings you partially or fully back to 7.4. So that's the way, at least in our hands for our studies, going to high altitude, you drive a change in carbon dioxide and the kidneys over time can then bring bicarb down further with a metabolic compensation. Look, we're halfway. I want to tell you the story of 2 expeditions before I tell you about a third. The first one is an incremental ascent to high altitude in Nepal. We then also went to White Mountain in California in 2019, where the time ago, 5 years ago. And then I want to finish off with a study we did last year in Nepal on ancestral lowlanders like me. Well, my ancestry is like Ireland, Britain, sea level, and Tibetan Highlanders who have an ancestry at say 400 to 4,500 meters. So this is the 2017 group. We made it to base camp after all of our studies. That, by the way, is Lintrin, and that glacier right there is a piece that broke off in the 2015 earthquake. Maybe some of you heard about that, but 7.8 magnitude earthquake happened nearby, broke off some of that glacier, washed across base camp and killed 19 people. Probably 10,000 people died in the Kathmandu Valley. We were a week away from departing. This was early May 2015, and I woke up on a Saturday morning to my phone exploding, like, did you hear about Nepal? Like, what? Turned out they had this terrible earthquake. They didn't need tourism, they needed aids, so we canceled our trip. Of course, our guide was like, you can still come. Come on, man. But he kept our money because we prepaid in good faith and we went back in 16, 17, and 18 for multiple expeditions. So here's the 17 group, the largest that I took over those 3 years. And there's the shadow of Lintrin, which is scary. The ice, you can see a bunch of tents there. The ice falls are just around the corner at the beginning of the Everest climb. So here's our expedition ascent profile. Did baseline measures either in Calgary or in Kathmandu, which is very similar at altitudes. That was the picture I showed you earlier. Up to Namche, one of my favorite villages in the whole world, this little shopping district at 3,400 meters. A couple days later up to Dibuche, which is not my favorite place in the world, up to Fairshé with that other picture I showed you from last year, where we took, and then up from Fairshé after our last measures, I headed all the way up to the last village that's a few hours away from Everest Base Camp. People don't live here year round, but it's the highest altitude village in the Nepal Himalaya in terms of places to stay before you head to Everest Base Camp and back again. I'll let you guess what we actually call it, given what it rhymes with, but it's a bit of a garbage town to stay in terms of accommodations. And so we did a bunch of arterial blood draws. I like using the iStats because they're portable. They were developed with Abbott in collaboration with the American military for remote stuff. I'd say radiometers are probably a better machine, but I find them difficult to service and get customer support for. I like the iStats. You can get cartridges in a week, and you can get any cartridge you want. So it's 1 cartridge at a time for 1 blood sample, and you can get any kind of assay you want to test. And so we brought a respiratory therapist along with us. Here she is taking arterial blood from a participant in Namche. And the poor Professor Broussard, who I tasked with doing the blood processing. That's what happens when you're a PI, you can tell other people what to do. Although he's more senior to me, but here he is set up in the bathroom lab processing some bloods. Believe me, I've done plenty of this too, so no one's above it. So we did these arterial blood draws. So just to give you a sense of how people responded to an incremental ascent where you never really have time to fully acclimatize and you stress the body again as you go up. Here's the O2 stimulus. So this is the atmospheric stressor and how it responded in the blood. You can see lots of variability in Calgary or Kathmandu. The big dot is the mean, and this is the scatter of all the participants. Asterisk means different from baseline. Dagger means different than previous altitude, just to give you an orientation for what's coming. So you can see 3,400 meters, 38, 43, not much different, all different from baseline, and then up to 52. I think there's something about 5,000 meters, you go off a cliff, and that's where people really get sick, largely because the oxygen levels drop really low. Some people down in the sort of low 30s. And here you can see, because they're breathing more to try to protect this would be worse if they weren't breathing more. They weren't mounting a hypoxic ventilator response. Because of that, they drop CO2. So there's our normal CO2 levels in Calgary or Kathmandu. You can see our 3 intermediate altitudes, and then lastly, heading down to 5,200 meters, a profound respiratory alkalosis. From here, we dump bicarb in urine. So that's, we're almost 24, but as I said before, look at this variability. Could be measurement error, could just be normal human variability. And then bicarb is dumped incrementally as you ascend to be pretty low, something like 18 or even lower than that at 5,200 meters. We saw numbers like that in Bolivia at 3,500 meters, which surprised me. But that's for us to sort out this semester with the student. And then lastly, arterial pH, how was it protected? Okay, statistically alkaline at 34, a couple days later at 38, they got back again. But then lastly, at 4,200 meters, they got alkalotic. And I can tell you from reading the literature, above about 4,300 meters, you're gonna be alkalotic forever. That might be a surprise that, the dogma in the biochemistry books say, you need to fix your pH. Your proteins will be all screwed up. Well, I don't know what to tell you. Every paper I can find above 4,300 meters or so, those participants, no matter how long, and I'll show you some from the top of Everest in a second, remain alkalotic forever. So shout out to the Canadian Sport Institute Alberta group who are here today, and I'm grateful you came. Collaborated with them a couple of years ago, and we wanted to utilize the data I just showed you from Nepal, where we did an incremental ascent, this is altitude, to an acute stimulation to the same inspired PO2 levels. So we just happened to serendipitously have arterial blood draws from Calgary, and then Calgary at a PiO2 of about 74 or so. And from this data I just showed you, Calgary to a PiO2 of about 76 or so. So we had a longitudinal 10-day ascent, and we had a 24-minute ascent by giving people a stepwise reduction in oxygen inspired in a lab. And then we took arterial blood draws here with an indwelling catheter with an RT and anesthetist helping, here with those single draws from the respiratory therapist that I showed you before. So let's just look at that real quick. So in terms of CO2, because you'd imagine the people in Nepal were acclimatizing, were breathing more, you can see that there's a double response in terms of dropping CO2, a larger renal stimulus to be sure. That larger renal stimulus led to a large dumping of bicarb. Not much happened here. This is just chemistry, we think. That's compensation. And then lastly, in terms of arterial pH and the change, you can see that they're more alkalotic from acute hypoxia than they are from sustained because they've dumped so much bicarb. However, I should say they're still alkalotic, as I said before, and I think it's just above that high altitude threshold that people will not compensate fully. This is under review with clinical science. I'm stoked and to share this with Monica by text earlier that it went to peer review. I can't believe it. So it's not rejected yet. So we made these Davenport diagrams. Remember to orient yourself, pH at 74, bicarb 24, CO2 of 40. There's our starting point. Look at the variability of this normal in Calgary. This is from the acute one. This is just over at the Winsport facility. And then you can see that after 24 minutes, dropping them down to a PiO2 of about 75 or so, you can see they've shifted along that non-bicarb buffer slope, about 3.5% reduction. So if you wanted to know how much chemistry is involved, just the elimination of CO2, dropping bicarb a bit, it's not much. From a high altitude perspective, there's normal in Calgary, Kathmandu, lots of variability again. And then after 10 days of ascent, not only have we moved to the right, but you can see we've dropped. So we've dumped a bunch of bicarb. It's about a 22% reduction in bicarb. But notice we still haven't gone back again because those people are still alkalotic. So I think it's helpful from a clinical perspective or from a research perspective to quantify the chemical component of drops in CO2 versus the longitudinal compensation of reductions in bicarb from renal excretion. Here's the pretty graph that we include in the paper. So you can see red is the acute, blue is the long-term 10-day acclimatization, sustained hypoxia, right on top of each other in Calgary. And you can see the difference between the groups there, 10 days versus 24 minutes, the same PiO2. And lastly, just to show you in one graph, 3.5% this way for the reduction of about 25% or so, 23%. Okay, let's head to a rapid ascent. This is a rapid ascent in residence to high altitude that we did in 2019. Here we are at the famous Barcroft lab in the Sierra Nevada mountains or nearby, close to the summit of White Mountain. 25 of us went up. There's my PhD supervisor, Richard, wearing his Crocs. We took a whole team up there to do some studies, baseline testing in my lab again. Flew to Vegas, behaved ourselves, drove 5 hours straight to the Barcroft lab to here, and spent 10 days in that Quonset, working in labs and sleeping in dorms. We did arterial blood draws there on day 2 and there on day 9. And then after that, we flew back down to Vegas and didn't behave ourselves. So just to give you a sense of this is the hypocapnia, normal in Calgary. You can see 2 days within 24 hours of arrival on day 2, we can see that they were hypocapic statistically. You can see that 9 days later, because they acclimatized, they're breathing more, and they're even further hypocapic. So there's the renal stimulus. Here's the renal response in terms of bicarb, reductions in bicarb, statistically up on 24 hours in. but even further dropped it on day 9. And lastly, the controlled variable. This surprised us. 3,800 meters, so not much of a stimulus, but higher than about the same height as Mount Robson near Jasper. And so you can see that statistically, they weren't different. So they fully compensated, at least statistically. But I think the important part here is the variability. And this is, I think, really important. We often swear at the variability. The big error bars makes us mad, particularly as trainees, but pay attention to that. That's the point. Some people really stayed alkalotic. Some people compensated, and some people overdid it, became acidotic. So there's lots of variability. And I think that problem with variability is worth embracing. If you have some response to all the things, you can see that we have a similar magnitude response here, but different levels of error bars that people often get upset about. But I think there's something meaningful there in terms of responders, non-responders, and breaking it up into groups, which would require a larger N to look at that. But so variability is the point, I think, and I saw it right in front of me when I printed these little chits off, that some people just didn't breathe much, so their CO2 didn't change much, so the kidneys didn't have to do anything. Some people had a really robust hypoxic sensor response. They had a really big reduction in CO2, but their kidneys had not yet compensated. They stayed alkalotic. And lastly, the third group that I noticed was there's people breathed a lot, they were hypocapnic, and then the kidneys kicked in and they brought their pH back to normal. Or in the case of these people here, overshot and become relatively acidotic. So some people overdo it. So acclimatization isn't always perfect and isn't always good. I think factors, and these are problems in high altitude studies, scent profile, time course of exposure, maximum altitude, rapid ascent to residence or incremental ascent, other acclimatization processes like hematological, you'd expect lots and lots of variability. And that inherent variability might be useful in helping us understand. So, as I said to you before, I hinted at this, that there's an altitude after which, or really a CO2 below which you will never compensate. So this is data from 4,300 meters. This is Bert Forrester, Jerry Dempsey, 5 people across a 10-day stay at 4,300 meters. Here's our stimulus. There's our baseline CO2 of 40. I should have said this over, so that's 40 there. That green thing is shifted right by accident. And then you can see over time at day 10, they're down to about 25 or so. So yeah, they become hypocapnic as expected. Okay? Then the response, they dump the bicarb from 24 down to about 18 or so. And then lastly, the controlled variables, the thing we care about, what is the pH starting just around 7.4 or so? Look, they become alkalotic and they never get better. They stay alkalotic for the whole 10 days. Now this was only in N of 5. Ainsley's group did it a bit better in 2022, where they went up to 4,300 meters in the Andes. Notice here, and I think there's 22 people or so. There's the drop in oxygen over time. So they did, Kelowna, 21 days. There's the drop in oxygen. So they're hypoxic, they're breathing more, they become more and more hypocapnic. So the process mean even more so than over here. They can see the drop in bicarb, but look, the pH, they're alkalotic and they stay alkalotic and never recover. So even as low as 4,300 meters, I think at least indigenous lowlanders, like many of us in the room, are walking around chronically alkalotic. So I'm doing a bit of a meta-analysis and systematic review on this. This is my first pass at it. This isn't the final graphs. I just did a quick survey of the literature to see if I was on the right track. I found 9 studies below 4,000 meters, just arbitrarily picked 4,000. Because of my observations in the literature, 15 studies above 4,000 meters. So you can see the altitude is different between the groups. Great. What about their PaCO2? As expected, the higher altitude studies had a lower PaCO2. Great. No surprise, a little bit of a bicarb. You can see a larger bicarb response in the higher altitude studies because they have a larger renal stimulus. Also no surprise. What about pH? I think I'm right. So there's persistent alkalosis in this group. They're higher than the group of studies below 4,000 meters. So I'll probably do a within subject comparison on this study. And I'm working with some other researchers to work on this now. So stay tuned for that study. The point here is that a lot of people think physiology is a dimmer switch. Sorry, a light switch. You turn it on, you turn it off. Response, no response. It's not that way at all. Every physiological response is probably a dimmer switch. You're set in the middle of the dynamic range from a muscle spindle up to breathing. You're in the middle of the range and you go one way or the other, but there's always going to be an asymptote below or above which you can't go any further. And that's what I think we're seeing here with this. I just want to show you before I finish how bad this could be. I've seen Michael Grocott and Dan Martin give versions of this talk over the years, but they did a remarkable thing and went with a huge commercially funded expedition to Everest Base Camp and wanted to put some clinicians on the top of Everest to try and get some blood gases up there. In '83, in the American Everest expedition, they got alveolar gas samples, but no one's, to my knowledge, has done arterial blood draws this high. So they wanted to get some arterial blood draws on the top of Everest at 8,900 meters or so. So off they went. On average, they only got 4 people and they only got it at 84. The weather was too bad on the summit, so they had to come down to the balcony at 8,400 meters and drop their snow pants and on each other got a femoral artery poke and took those samples, put them on ice, ran down to Camp 4 where the blood gas analyzers were. I'm sure there's no measuring error there. And got PaO2 25 on average. What's normal? 100. Walking around, having just exercised on the top of Everest, these are conscious humans with a PaO2 of 25. And they're breathing so much, normal's 40, remember, right? CO2 is 13 on average. I don't know, that's gonna cause so much cerebrovascular constriction. I can't believe they're conscious. And yet their CO2 is very low. So let's look at some data. So here's their ascent profile. I've been as high as this. Actually, I've been higher at a summit nearby to take this picture at 50, a picture like this at 5,600 meters. But basically 53 is the end of the Everest Base Camp. And then you head up the ice falls to Camp 1 at 6,000 meters or so, Camp 2, 3, 4, on the col between the Lhotse face and Everest, up to the summit, came back down to the balcony at 84 and got these measurements. And so this was like 70 days from the UK when they got these measurements. So pretty long study. I don't want to spend too much of time on this. Look, you can look at O2, oxygen saturation, hemoglobin concentration, and oxygen content. And you can see here that content is pretty well maintained, actually. Not until you get up to 8,400 meters is your content really down. But again, they only have 84, four people at the top there. Here's the thing I want to show you. Here's their table. I put in normal on the right-hand side for your comparison. So look at your pH. On average, 7.53. Remember level 7.4? Profound respiratory alkalosis that even after 70 days, they're not getting better. O2, they said 25. PaCO2, 13. Some people are down to 10. Their bicarb, normal's 24. They dumped down to 10. It's the lowest bicarb I've ever seen, short of the chamber study in the 80s where they did a simulated Everest climb. They had similar like 11, something like that. So that's crazy low. I saw one person have this in Bolivia at 11. So that's diarrhea. That's another conversation. And oxygen saturation, 54 on average. You know, you're all 85 to 90 right now. Just to give you a sense of how bad it can actually be and you're still conscious. So what did we learn before I wrap up? You learned that you get a ventilation-mediated hypocapnia, you get a respiratory alkalosis, call that chemistry, a small drop in bicarb, only because of the loss of CO2. Over time, the literature, as far as I can tell, looks like between 3 and 6 hours is the earliest time course it starts. You start dumping bicarb, within 3 and 6 hours, bringing pH back to normal, unless the hypocampic stimulus is larger than sort of these mild ones. So anything over about 4,000 meters, it looks to me like people are walking around alkalotic. And there's tons of variability, as you saw before. Let me finish. I've got just under 10 minutes, and I just want to show you some new data. Consider leaving this out and stopping here, but I think this is sexy, and I want to show it to you. And it's good practice to talk about it, and I'm curious if you have some feedback. It's in review at PNAS. It hasn't been rejected yet. So watch, I'll check my phone after this. It's like, sorry, this is a garbage study, go away. Anyway, so this is the genetic adaptation side of things. These are the sort of general locations, as I mentioned earlier, where people live at high altitude. Ethiopian highlanders are difficult to study and they're quite remote, so less work has been done on them. The Andean highlanders is a very rich literature. The Tibetan highlanders are also a rich literature. The name Sherpa is not a job description, it's an ethnic group. They are Tibetan highlanders that just happen to cross for them, an artificial border between Tibet and Nepal. They're Nepali, but their religion, their culture is Tibetan Highlander. And so the Sherpa happen to be very good at living and working at high altitude and end up working in tourism because they're so incredible at dealing with high altitude and exercising, but it's because they're Tibetan Highlanders who live there for millennia. And then there's, of course, mountains just down the road, but we're not living very high. So the Tibetan Plateau is often called the roof of the world. It's huge, 2.5 million square kilometers. Average elevation is higher than 4,000 meters, and that's the altitude that I'm gonna go to in a second, is about 4,300 meters. And that plateau has probably been occupied through archaeology as long as 40,000 years ago, but not permanently settled. And this is a hot, I mean, anthropology is hard. I'm glad I'm not doing this work. Just making this slide took me hours trying to read all these different papers that were arguing with each other. But maybe 6,500 years ago, people were living there chronically or had permanent settlements. There's a nice review by Gilbert Kawai in 2014 that talks about all the sort of known, this is a decade ago though, all the known physiology of Sherpa and what makes Tibetan Highlanders, makes them special at altitude. If you were to put a gun to my head at a conference, I'm not sure I could tell you definitively what it would be. A lot of it's biochemical, maybe some blood flow differences. The dogma, and Martin will know this, the dogma is that the hemoglobin concentration is hemodiluted. In my hands, and a recent study that I reviewed shows it may not be the case, at least early on, that they have similar plasma volume and hemoglobin concentration to lowlanders, but over time it's been showed acclimatized high altitude natives, Sherpa Highlanders have a hemodiluted perspective there. But what's missing from this is the kidneys. What the hell? But when you see a gap like that, you gotta think to yourself, I might be onto something. So why do they do so well? Not much is known about the kidneys. In a recent paper where we combined a bunch of arterial blood draws together, a bunch of labs led by my old student, Mike Timko, showed that there is a differential renal response between Tibetan Highlanders and lowlanders, but these are acclimatized Tibetan Highlanders, and so that's the caveat here. Here's one of the figures. They are lower in pH than the lowlanders, but this is a convenient sample of Tibetan Highlanders recruited, living chronically at high altitude, and then these lowlanders were kind of brought up on various timescales. So this wasn't a well-done study. It was really just a combination of a bunch of blood gas data that we all put together. So let's do it right, shall we? Let's take some people of Tibetan Highlander ancestry, recruit them in Kathmandu, bring a group of lowlanders there, recruit them voluntarily, and then go up together and test them in Kathmandu and test them at 4,300 meters at the same time course of exposure, knowing that 4,300 meters is around the threshold for lowlanders like me to not compensate, to not get there. And so we hypothesized that Tibetan Highlanders would be better at it. We recruited a bunch of lowlanders and Tibetan Highlanders in Kathmandu. These are just university kids, like studying finance in Kathmandu University. So a lot of them hadn't been up high. They just happen to have the last name Sherpa and have Tibetan ancestry as part of the family, as evidenced by anthropological interviews by our team member. We did capillary blood draws, which are thought to be, from meta-analyses I've read, a good surrogate for arterial, for CO2 only, not for oxygen or garbage, but for CO2 and bicarbonate pH. They're probably adequate. Used a radiometer, and Martin was kind enough to give us some capillary adapters that made the study possible. So now you get to see the fruits of your kindness. And so 15 and 14, roughly equal sexes. We had a 15th, but her hematocrit was too low and she was anemic, and so we couldn't include her. And then up we went and just took the main metrics that we've talked about. Here's the ascent profile. There's Nick and I about to get into a helicopter to head up to the village, and here's my student Rodian taking capillary samples on one of our participants. It's in review now. So just to show you, look, both groups hypocapnic with ascent. Both groups ventilate more with ascent, making them hypocapnic. Both groups have are acclimatized to some metric I don't want to bore you with today, but you can see the breathing is all up. They dump bicarb, both groups. There's already a hint there what's going on. Their basic success is also down. This group is alkalotic. This is the lowlander group. The Sherpa are recovered statistically. So there's the big finding there that I think hasn't been shown before. And so that's all the boring stuff around ventilation, but look, they're different at everything. Their CO2 is lower, their ventilation's up, their chemoreflexes are up, their bicarb's lower, their basic success is lower, and you have a base deficit, and their pH is perfectly compensated statistically. And so that's the first demonstration that I know of an incremental ascent profile. You can see on this little graph we made here of this index of renal reactivity against changes in pH. They're slid down, they're closer to normal pH, and a percent pH calculation that Jerry Dempsey told me to try, they're also different as well. So every way you look at them, Davenport diagram on top of each other in Kathmandu, different in Fairshade at 4,300 meters. So I think we have something really cool there about Highlanders. So my last slide, the kidney is important. You know, as a respiratory guy, I'm embarrassed to say, maybe breathing doesn't matter as much. You know, we know the kidneys are important in terms of EPO release, acting on bone marrow, make more red cells, make more hemoglobin mass, protect your oxygenation, great. So ventilation of the kidneys acts together and the bone marrow acts together. Sherpa have a similar EPO and hemoglobin mass response, at least in our hands. I showed that at CSEP last year, and I was gonna show it again today, but I didn't want to bore a few of you that were there. So that's this new talk. We showed a more rapid and larger renal response in Sherpa compared to lowlanders. And I think it suggests selective pressure anthropology that these Tibetan Highlanders evolved at 44 to 4,500 meters over millennia, and thus they're adapted to it. And that's long-term genetic adaptation. Is the kidney the most important organ for high altitude colonization? Maybe. I should thank many of our participants. Here's a bunch of the Sherpa. The girls wouldn't let the boys sit with them at this coffee shop in Namche on the way down. But some of our team, as well as some of our Sherpa participants, are here waving hello. What a lovely privilege to get to know a bunch of them with this incremental ascent. Looks like I've crossed the finish line just on time. Thanks to National Science Foundation and INSERC for funding this work, and you for coming and listening so attentively. Thanks. Too long.
We're looking for questions.
I'll stick around though if you have questions. I'm happy to stay.
Very nice and impressive presentation and studies, that's for sure.
Thanks.
So I'm wondering about the pH. So the pH measured is in the blood.
That's correct. These were
Right.
either arterial or capillary blood draws, yeah.
So it's basically extracellular. I'm not sure about capillary and the extracellular matrix of the cell versus also the intracellular, like the inside the cell.
Yeah.
Because I'm sure there are buffer systems around the cell to make the proteins in the membrane like in the right shape and inside as well. So what do you think about these?
Yeah, that's a great question. I love that question. It's something I've been thinking a lot about, that, you know, much like when we measure blood glucose, who cares about blood glucose? It's intracellular glucose that matters. If insulin is working to insert glute transporters to get it into the cell, is actually what matters, right? And so similarly, the cell cares about what's around it directly, but also what it can control on the inside. So, you know, extracellular fluid, the blood is only one of two compartments. We're measuring a surrogate. So it's the best we can do. It's the gold standard measurement clinically to measure blood gases, blood pH. But I agree with you that I don't know what's going on inside a cell. So, you know, when I say people are walking around oncolytic, they are in blood, but who knows what cells are doing? They're probably just fine, or else we wouldn't be walking around. I think that's your point, right? We don't know. The actual compartment that matters is probably the extracellular fluid compartment around cells, but likely intracellular as well. So now I need to do animal studies, so thanks a lot. But I agree. Yeah, I don't know what to say other than the fact that this is what humans can measure and what we care about clinically. But I think you're right. That's a caveat we have to think about. Yeah, thanks for that.
But you think it said that there are other systems that would regulate the pH inside the cell and
Yeah.
bring it back to what the cell likes.
Yeah, we don't have blood proteins in the intracellular fluid space, and so it's a lot more susceptible to, especially if you're dumping bicarb. All you have in the brain, cellular fluid compartment and tissue everywhere is bicarb, because you don't have the blood proteins. So and then when you start dumping bicarb, I think the extracellular fluid compartment probably also is alkaline, because CO2 just diffuses everywhere. Bicarb's lower, you probably are alkaline, but the cell itself can probably maintain its own intracellular environment. Again, I don't know. How do you get that evidence? How do we have to do cultured cells? Probably. I haven't read that literature, but I appreciate the prompt to do so. Yeah, it's a good thought. Thanks. Yeah, hi.
Hello, I found this presentation really interesting. Like, it's really connected to one of the physiology classes that we take before in the PhD degree. But I have one quick question. So for your results, there you have like a massive deviation between the concentrations of the bicarbonate concentration and the partial pressure of carbon dioxide in the blood. How do you account for like what the individuals are doing while they're up there in the altitude? Is it like always they're doing the same activity, or some of them might be more physically active?
So you're asking about the lifestyle.
Yeah.
Yeah, that's a good question. That's a major caveat that we get called on and review over time for our incremental ascent models. Like sometimes you're trekking 7 hours the day before. some of us don't do that every day. And so that's a massive stressor that might cause dehydration. I can tell you that the osmolality measures, at least on all these participants, were constant. So they were, and because we encourage people to drink water, and so they probably weren't dehydrated. In Bolivia, we had people with increasing creatinine from their diarrhea, so they probably, some of those were dehydrated. I didn't get a chance to show you Bolivia data from July this last year. So I think it's a major caveat though, like, do they have Travis diarrhea? Are they exercising and sweating a lot? These are things we have to start thinking about from a fluid electrolyte balance perspective. Because Stuart would say bicarb is not the only answer for pH, that maybe the strong ion difference is important, and that includes other ions. So I take your point, but it's something I worry about a lot, and starting to think about other ways of looking at fluid electrolyte balance. But yeah, the incremental ascent, they were exercising. But their osmolality was fine, so it's probably okay. I think, you know, to be honest, these high altitude studies never report things like our participants talked about diarrhea. So we're gonna do it with our Bolivia study. We're gonna look at those that did and those that didn't, and there may be a difference. They're dumping bicarb out of their butt instead of the kidneys. So I think if you're interested in acid base, there's a caveat there we need to worry about. I think we need to be more honest about this kind of stuff, so thanks for that. Yeah, hi.
Hi, you mentioned that patients become alcoholic beyond a certain altitude, about like 4,000 meters, and they develop like persistent respiratory alkalosis.
Yeah.
And I may have, yeah, and I may have missed the point, but does it still remain, like, do they remain in an alcoholic state even when they return to, let's say, sea level altitude?
Right, that's a great question. So in my data and my reading of every paper I can find above 4,000 meters suggests that lowlanders are remain alcoholic for as long as the studies go on. I mean, you know, 6-month studies haven't been done, but we should do those. We should go to 5,300 meters and spend 6 months there. Who wants to go? To a mining town in the Andes. But to your other question, washout data hasn't really been done well in these kinds of studies. There's time course of decarbonization. I haven't read deeply enough about it, so I'm embarrassed to admit that I don't know of a lot of papers. So I haven't been systematic about it, but I don't think it's been done very well. Watching the time course of bringing the hemoglobin mass back to normal again, ventilation, CO2, acid base, I don't know. I think as soon as you remove the hypoxic stressor, your CO2 is gonna come back up. You'll probably fix your acid base again, but I want to build that in, and we've been talking about it, build that into a future study to actually watch the washout over a couple of days upon return. It's a great idea, but I don't think it's been well done in the literature. So I wish I had more for you. Yeah, hi.
Hey, great talk, very engaging, thank you. I don't have a huge background in physiology, but I was wondering about sort of the effect of training.
Yeah.
Which I didn't, see it all in yours and was curious
You're
about.
right.
I'm curious what sort of strategy differences, like pathways to acclimatization you would see in somebody who you took from like couch to base camp, as opposed to like triathlete or, Ironman to, base camp, and how that would maybe be different to like the Highlanders.
Yeah, that's a great question. When you say training, you don't mean altitude training, you mean training like exercise
Yeah, say
training.
like a well-trained endurance athlete at sea level.
Yeah, my reading of literature suggests that susceptibility to mountain sickness at least is not dependent upon fitness. Certainly your ability to exercise at altitude is probably better than mine, because you're probably fitter than me and you're younger than me. So I think the ability to train or exercise at altitude is affected by fitness. I think that's uncontroversial. But whether or not you're susceptible to sickness, to mountain sickness, which includes symptoms like headache, dizziness, lightheadedness, fatigue, weakness, gastrointestinal symptoms. They used to think sleep, but now they don't, right, Martin? But everyone can sleep apnea up there, which is another conversation around altitude training camps that we can talk about. I don't know literature, but I think we have an expert here about at least AMS around fitness. I don't think it's been shown that there's no effect on sex, age, or fitness on susceptibility. Is that true, Martin? Yeah. I think what matters more, at least up to my experience, up to 5,000 meters, it's all behavioral. It's fluid intake, it's food, like you get anorexia, but you gotta eat. It's ascent profile, it's exertion during exercise. And here's the thing, the young fitty fits, like guys like you on my trip get sick because you're used to being in front of everyone. And so they push really, I'm not saying you want, I don't need to, but no, I need to actually. But it's the young fit guys and girls that are used to being in front, they just push really hard. And then a day later, they're sitting them down because they're sick. And so I think the exertion's a big part of it. So the ability to exert's there if you're fit, but I wouldn't do it if I was you. So to making sure your ascent profiles 400 meters per day, take a rest day every 2 or 3 days or so. I think it's all behavioral up to 5,000. After 5,000, I think it's a roll of the dice genetically. That some people are just susceptible and some aren't. I've sent, first 2 years I was in Nepal, we sent people down with pulmonary edema and they were both young men. Who knows? So I wish I had more for you around fitness. I don't have a background in training, so we're coming at it from different angles, but I think the incremental ascent model, that may play into it a bit to some extent, because like in 2017, I was a little fatter than I am now, believe it or not. And I had a hard time with that, because other people are like, this is easy, so.
Actually, a follow-up to that, question, have you seen a difference in someone that's been there at their first trip versus someone that's going there for the third time?
That's a great question. There was a paper in, was it Nature that talked about red cells perhaps retaining some colonization? Anyone know this paper? But I don't, yeah, can you tell us? I'm glad you came, Martin. You wanna come on up and put the microphone on?
There's some like biochemical holdover from previous ascent. I think it's a Rob Roach study. But it's pretty, that was like, go up, go down, wait a few weeks, go back up. It wasn't like an annual trip or years apart. It was more like they were still acclimatized.
Yeah.
There's some minor pathways, but
So they weren't fully declimatized, yeah. I mean, Rob Roach's stuff is probably the best place to look for some of that declimatization data that I was talking about. Well, first of all, the answer is I don't know. But I think there's probably some epigenetic changes, and we did some work on that in 2018 that we published. That's less outside my expertise. I just happened to organize the expedition and, got sort of like involved in that work a little bit. But I don't know a lot about that background, but I think there's some epigenetic changes that I didn't talk about with my continuum that probably persist. There's evidence that people that have famines, then their kids are susceptible to obesity, like there's something going on there that's not genetic in the purest sense that we're used to. So maybe, but I think that's a brand new field that's, still growing. But I've been like, sorry, just saying, I went 2016, 17, 18, same exact descent profile, same exact place. And, maybe fitter some years than others, but didn't really notice any differences in mountain sickness or anything between the years myself. That's just anecdotal, so.
Art,
You were talking about variability and you showed a case where people overcompensated.
Yeah.
And got acidic.
Went too far.
What happens over a longer time at that same altitude? Do they come back or is that a steady?
Good question. I'd have to look at those participants to see, because we connected the dots for once on that. And so I was gonna talk about them. Forgive me, give me a second here. Let's go find that. Because we connected the dots so we might see it. Oh, there we go. So these ones here, look at this. So I think that was a rapid response. These people actually came back up again. So I think it was just the dimmer switch went too far, and then they kind of like 8 days later were fine again. So, we don't have, this isn't a big end, this was 21 people, not even enough to do a sex difference, which is studies I'm working on. So I think there is probably an acute versus chronic effect as well, but that's a great question, that we should be looking at some of that. Because I think there's a difference between acute responses and long-term responses.
So you were saying that if you were able to do a study for 6 months, you'd expect their pH to still be apoplodic. Say you were able to do one for years, do you think their response would ever look like the Tibetan Highlanders and like come back down to a normal pH?
I mean, if it's true, then only that we would still call that acclimatization, that's not genetic.
Yeah.
So I don't know, I think all bets are off, like I have no idea. The longest studies I've seen, 70 days with the UK one on Everest, that's probably the highest and longest one I know of. I know Phil Ainsley wants to do a 6-month trip to Cerro de Pasco at 5,300 meters, that's the study I want to go on. And test people early and late after 6 months, but I think that's the big unknown question. And I get it in every talk, like you're talking, your question's great, like I'd like to know, how long would it take for them to get back to normal, if they will at all?
Exactly.
Because to your point, like, can you really be, people have challenged me, a Bolivian researcher I was just talking with in Bolivia in July, I was like, surely they're not walking around alkaline all the time, it's like, look at these studies, like, look at my meta-analysis, like, they're all alkalonic. And so intracellularly, they're probably fine, but at least from a blood perspective. So I don't know, I'd love to see that time course to see if they finally do correct. What altitude and how long would it take back to normal or not? So I don't know the answer. I'm not even sure I'm willing to speculate,
Yeah,
whether I'd say, yeah, no, they stay that way or anything back to normal. I have no reason to know either way.
Yeah.
It's a good question. So let's do that study. Can we organize that study? Yeah. Oh, yeah, Martin was before, so
Thanks for the great talk, Trevor.
thanks for coming.
Anytime. The variability is really interesting. I like that you highlighted the point that you should be against variability. It's part of the process, but what do you think the consequences of it are? So when people are on either ends of this, what functional outcomes does it have for their time at altitude or their health, their performance? Do you think it's meaningful that way?
Don't know. That's why I think it's worth asking. Like, my point here is that it may be instructive. I mean, look at me waving my hands here, like at random, like, this may be instructive to understand altitudes. I don't know. The fact that some people overdid it. I, like you, I think that AMS is a subjective qualitative metric that may not be best served by statistics, or statistical comparisons. But, so how else do we get at it? Those that overcompensated and were acidotic, are they better because they're not breathing more? I don't know. I'd love to dig into that more, but I think the best study to do this right is to go up to some altitude multiple times and get a huge N. I think we need like 20 or 30 per group here to do it right. And then we can think a little bit about, on the same time course, try and control for other variables like perhaps fitness, perhaps diet. Eric explained some things that should be giving people ensure to drink every day, and that's all they should be eating. It's like, well, participant buy-in is important in these studies. But I think the best study then is to do this properly, get a handle on these different phenotypes and the variability of these different phenotypes and see if there's some sort of functional outcome, whether it's exercise performance, this is where I need you guys, because that's not my forte, or acute mouth sickness, or I don't know, some other metric of health. It's a great question. I don't know what the implication is of it. Maybe it's just the way it is. Or it's measurement error, blood gas machines may or may not work well.
Sure. I mean, I think with the lead score, not your problem, but the problem with it is it's taking so many things and putting them in one score. And
Yeah.
if you have something that's kidney based, and then you're adding somebody's fatigue, their nausea, all these things that are multifactorial, it's never gonna correlate with a really specific measure. Or it's unlikely to, because they have different origins. So I think getting away from that. Yeah, I tried my best to kill it, but it's still there.
It's hard, yeah. Well, anytime you go to developing country, you're gonna have Travis diarrhea. What I've heard it said, in Nepal, there's two states of being, you either have Travis diarrhea or you're about to get Travis diarrhea. Probably both. We felt that in Nepal, in Bolivia too, like you just can't escape it. So I think when we went to White Mountain, those data were from White Mountain in California, no Travis diarrhea. So I think we need to maybe perhaps stay on the continental, North America.
Yeah, for sure.
For some of these questions, to try and eliminate some of those other variables like diet, like nausea, vomiting, diarrhea, activity like drive to a lab instead, those kinds of things. So I like the point. We need to try harder. It's sexy to go to Nepal, right? But I admit that those kinds of papers, we never talk about those caveats enough, because I think they're affecting our data. Thanks.
Let me just return to that thought again.
Oh, no, I think it's time to go.
Let's say we don't think there's a problem with measurements. What happens between day 0 and day 2 for those weird people who overshot? Did they only go down, or do they perhaps go up and then too far down?
I love your question. Every study I can find, because of logistics, you can only measure what you can measure. So we landed in Vegas on a Sunday afternoon.
Well,
And
so also, I'm not asking you as a fact, I'm just asking you to speculate, what do you think happened between 0 and 2?
don't know, there's only one study I know that did an early measurement that I can't forget the details of it. So I don't think that's been well done. Like we drove up over 6 hours to the lab and then started testing the next morning at 7 AM. And then kind of did art draws all day. But within 24 hours, these measurements were taken on day 2. So all I can say is within 24 hours of acute arrival, rapid ascent and acute arrival at 38, that's what they look like. What did they look like that night? Those are the studies I want to do a better job of. Let's do an art draw upon arrival. And let's answer that resolution question a bit. That's where chamber studies can come in handy. If you want to do short-term, high resolution effects, I'd like to do that more. So I wish I had an answer for you. All I can tell you is that 24 hours later, either measurement error or normal physiology, they become relatively acidotic to their normal. We didn't do delta, we didn't study delta responses here, maybe we should have, so. But it's probably an unsatisfying answer. I agree with you, it's important and interesting, I just haven't done it. I haven't seen it done.
Maybe time for one more question? Yes, so the adaptation, genetic adaptation that you talked about at the end. So how do you think about it? So where is it happening? What are the mechanisms? Like the pH is going down. How is it going down?
I think that. It's a good question. So this is not acclimatization. Well, sorry, it's genetics affecting acclimatization. That piece of it, and this is like, I wanna underscore it enough. These were 22-year-old kids going to university in Kathmandu that may or may not have been up high ever. They just happen to have ancestry, like I have Irish ancestors. They have ancestors that for thousands of years lived at 4,000, 4,500 meters. I think there's selective pressure on that population to do better with acid base while living at that altitude. All I can tell you is that they acclimatize better over 7 or 8 days. That's all I can tell you. And then we have data over, when they're acclimatized, they were probably still alkalotic in a larger set, immune sample. But, there's been lots of work recently on genes implicated in oxygenation and control of oxygenation and protection of oxygenation. EPAS1, for instance, running in the HIF pathway, something around there that probably leads to carbonic anhydrase expression is my guess. But again, I don't know. So we had geneticist Abby Bigham from UCLA with us on that NSF grant. We pulled venous draws at everybody. And so we were looking for splenic responses. And then I threw a wrench in a spanner in the wheel and showed acid base changes in the kidney effects. And now she's gonna start chasing this a little bit. I don't know how you chase kidney genes, but I think there are genes related to oxygenation that are probably different. And maybe to do with expression of carbonic anhydrase. That's where I think we should be looking.
So do you think that kidneys, they do a better job at eliminating the bicarbonate?
That's what we saw. Yeah. Sorry, I know I should have taken out, there's a bunch of shit I put in there, I should have put in there. My bad, stand by, almost there, promise, hang in there, thanks. There's bicarb. There's the lowlanders, Kathmandu versus Fairshade, 4,300 meters on day 8, 9. Same time course of ascent. There's the Sherpa, they're already lower. That's something I didn't talk about. The Sherpa looked to be lower from a CO2 and bicarb perspective in Kathmandu. And so the delta responses are affected by the fact that the short impulse is different. So I think they were acclimatized differently at 1,400 meters, which is a little higher than Calgary. So I mean, how do you assess that? But look at this value, like lower than here. So I think even the delta response was larger, even though they started lower. So yeah, I think they had more ability to excrete bicarb, probably because of perhaps carbonic anhydrase not working as well. I don't know. Is there the cellular stuff? I like the cellular questions. It's where I started, but it's something I need to do some more reading about from animal models around kidney tubules and that kind of stuff. But
Great,
thanks.
we'll follow there and let's thank Dr. Derek for this.